Let's move to the next topic, female reproductive part. Previously you learned detailed about the structure of androecium, that is the male reproductive part of the flower. Today we are going to study the female reproductive part, that is gynoecium. Gynoecium, which represents as a female part of the flower. It refers to a pistil. Pistil consists of stigma, style and ovary. Stigma, style and ovary. In the ovary, the ovules are present. These are ovules. Stigma is the region where the pollen grains are landing surface. This is the area of landing region of the pollen grains. The pollen grains are dusted on the stigmatic surface. So it's a landing region of pollen grains. Style is long, slender region just beneath the stigma. Just beneath the stigma there is a slender, long region called as style. The basal swollen part, this part, the basal swollen part is called as ovary. Within this the ovules are present. The ovules may be single or more. There are single ovules or many ovules are present within the ovary. Let's study the structure of ovule. Structure of ovule. Structure of ovule. The ovule, which is otherwise called megasporangium. Megasporangium. The megasporangium or this ovule, which is made up of a stalk and the body. A stalk and the body. So this is the body part and there is a stalk which covers with envelope called integuments. There are one integument or two integuments are there. Integuments, either one integument or more than one. So this integuments completely covers the body. So this part is the stalk. The name of the stalk is funicle or funiculus. Funicle or funiculus is the stalk of the ovule. This is the structure of the ovule. The structure consists of a stalk and the body. The stalk of the ovule is named as funicle or funiculus. There is a region which connects the stalk with the body of the ovule is called hilum. This is the region which connects the stalk with the ovule, with the body of the ovule. Stalk is connected with the body of the ovule by a region is named as hilum. And in the inverted ovule, this ovule is, the structure of the ovule is vary. This is the completely inverted ovule. In the inverted ovule, the stalk or the funicle which merge the body of the ovule and form to merge the body to the funicle, there is a long ridge. So this ridge is known as raphe. and this is the center of the ovule. The center of the ovule is parenchymatous cell. This region is parenchymatous cell. Parenchymatous cells. It contains the reserve food. The name of this part is nucellus. Nucellus. So this center region is completely made up of parenchyma cells with a reserve food, storage reserve food. So it's called nucellus. The nucellus, which is covered by envelope. There are one envelope or two envelopes respectively. So these envelopes are known as integuments. Integuments. This is the outer integument and this is the inner integument. Here two integuments are present. The integuments are not completely covered the entire ovule. This is the top region and this is the basal region. In the inverted ovule, this is the top. So it leaves the integuments which leaves at the top region, and this opening, it leaves a opening. That opening is known as micropyle. There is an opening which is called as micropyle. Just above this, just opposite to this micropyle, or this region where the integuments, the nucellus are placed there, in this region just opposite to the micropyle is called as chalasa. Chalasa. So this region is chalasa. Actually this region is called as basal region, and this region is the top region, top and the basal region. So just opposite to the micropyle there is a region which is called as chalasa, and inside there is a sac-like structure. Towards the micropyle, within the nucellus, there is a sac-like structure. It's called as embryo sac. This sac-like structure is called as embryo sac. Embryo sac. So in the structure of ovule, these are the parts. When you are learning this question, draw this diagram. First you learn all the parts. First part is funicle. Funicle is the stalk of the ovule. The ovule is otherwise called megasporangium. So in the megasporangium or this ovule, the stalk and the body, it's made up of a stalk and the body. The stalk is called as funicle or funiculus, and there is a region, hilum, which connects the stalk with the body of the ovule, and it is covered by integuments. The envelopes or the coverings, they are called as integuments, outer integument and the inner integuments. With that, if this outer and inner integument are the two integuments, when it is present single integument or two integuments respectively, which is called as unitegmic ovule or bitegmic ovule. Based on the number of integuments, the ovule is classified into unitegmic or bitegmic. If single integument is there, it's called as unitegmic. If two integuments are there, it's called as bitegmic. the unitegmic or bitegmic ovule. And based on the position of the sporogenous tissue, nature of the sporogenous tissue, there are two types of ovule, teninucellate, crassinucellate. Teninucellate and crassinucellate. There are two types of ovule based on the sporogenous tissue. The sporogenous tissue which is present in the hypodermal region is called as this teninucellate. When it is present in a subhypodermal region is called as crassinucellate. And there is a region, this is the basal region and this is the top region. In this region there is a group of tissue present in between the chalasa and the embryo sac. There are a group of tissue present in between the embryo sac, this is the embryo sac and the chalasa is called hypostase. And there are a group of cells which present in between the micropylar region and the embryo sac is called epistase. So, epistase and hypostase. So, here there are all this part of this ovule is 2 mark question. What is micropyle, what is funicle, hilum, raphe, embryo sac, nucellus, integuments, chalasa. It reveals the structure of ovule or it is known as megasporangium. And there are different types of ovule. Based on the orientation, form and position, there are different types of ovule. They are classified. There are mainly 6 different types of ovule. 4 main types and in addition to the 2 more types of ovule. They are called as orthotropous ovule, anatropous ovule, hemianatropous ovule, campylotropous ovule. These 4 are the main types. What are they? Orthotropous, anatropous, hemianatropous, campylotropous. In addition, there are 2 more types of ovule. They are amphitropous ovule and succinotropous ovule. Let us see how it is orthotropous. They are based on this classification of the ovule is based on the orientation, form and position. So, this type of ovule is orthotropous. The structure, these are the structures of different types of ovule. Here orthotropous ovule. The orthotropous ovule, the presence of micropyle. This region is the micropylar region, opposite region is chalasa. The micropyle of this region is in the distal end of the center. Distal end is away from the center. So, this is the center part. The micropyle is present away from the center or distal end. And the position of this micropyle, chalasa and the stalk funicle, which comes as a vertical region. It is positioned or it is arranged in a straight or vertical position. So, this type of arrangement is called orthotropous ovule. What is orthotropous ovule? Orthotropous ovule, the micropylar region is distal end of the ovule and the position of micropyle, chalasa and the funicle as a straight line, which lies as a straight line or a vertical line, is orthotropous. Then anatropous. Second type of ovule is called as anatropous. This is the completely inverted shape of ovule. Most of the dicots and monocots is shown this inverted shape of ovule, that is anatropous. The name of the ovule is anatropous. So, because of this inverted nature, the position of the micropyle, chalasa, micropyle and the funicle. This is the micropyle and this is a stalk funicle, which is lie closer to each other. So, this is inverted ovule, anatropous ovule. And the third type of ovule is named as hemianatropous ovule. Hemianatropous ovule is placed as a transverse position. This position is placed as a transverse position is hemianatropous ovule. So, hemianatropous ovule, here also the micropyle and this micropylar region and the funicle which is adjacent to each other, and the position is transverse in position, hemianatropous. Then campylotropous ovule. Campylotropous ovule, the embryo sac is bean shaped or curved shape. The shape of this is bean or curved shape, and here the position of micropyle, funicle and the chalasa which is very closer. So, this type of bean shaped embryo sac or the shape of this curved nature is called as campylotropous ovule. Then these four, orthotropous, anatropous, hemianatropous and campylotropous, are the four main types of ovule. Apart from this, there are two more types of ovule. They are amphitropous ovule and succinotropous ovule. In amphitropous ovule, here the nature of this embryo sac is curved and it looks like a horseshoe shaped structure. Because of the curved nature, it shows as a horseshoe shaped structure. So, here this is the micropylar region, the micropyle and the funicle and the hilum all are very close to each other. Hilum, stalk, that is funicle, and the micropyle is very close to each other. Then another type of ovule is called as succinotropous ovule. In succinotropous ovule, the funicle, the stalk of the ovule is very long. So, it may be seen in cactus plants, Cactaceae family. This funicle is very long. So, this type of ovule is found in cactus plants. You know that cactus, that is xerophytes. So, succinotropous ovule. So, this question is types of ovule. Today you learn two questions. One is structure of an ovule, another one is types of ovule. Both the diagrams are important. Structure of ovule, you draw this diagram neatly and label all the parts. Learn each and every part as a 2-mark question. So, here how many points are there? There are integument. First part is funicle 1, hilum 2, 3, 4, integuments 5, nucellus 6, embryo sac 7, micropyle 8, chalasa. So, this all questions are 2-mark questions. Learn one by one. What is funicle? What is micropyle? What are integuments? Hilum, raft, embryo sac, nucellus and chalasa. In addition, what is epistase? What is hypostase? What is unitegument, bitegment? Then what is teninucellate and crassinucellate? And here orthotropous ovule, anatropous ovule, types of ovule. There are 6 different types of ovule with diagram: orthotropous, anatropous, hemianatropous, campylotropous, amphitropous and succinotropous. Hope that you understand these 2 questions very clear. Before the next class, you learn these 2 questions and complete the diagrams. Thank you.